Ma'am, we are live. Bharti, ma'am.
Yes. Good morning to all the dignitaries and the participants joining us today for the fifth session of our webinar series on fermentation technology. On behalf of the management of Guru Nanak College, Department of Biotechnology, I take an immense pleasure in welcoming all the participants and dignitaries to the session 5 uh, of this webinar series. It's my proud privilege to kindly request our Dean Science, Dr. Noor Jahan ma'am, to deliver our felicitation address.
Thank you, Bharati ma'am. Respected Principal Dr. N.G. Raghunathan, General Secretary and Correspondent Sardar Manjit Singh Nair, Vice Principal Shift 1 and Shift 2, and Dr. Veena Gayatri, the resource person of today's webinar on metabolite production and mass transfer, and deans of various schools, head staff and students, especially from biotechnology department, and students and staff from various other institutions. My warm welcome you all. Okay. Actually, see, we had the privilege of listening to Madam. Our Guru Nanak Center for Research already had a webinar. Thank you, ma'am. Thank you for that wonderful webinar. Here also, we expect a lot from you, and this is an opportunity to listen to your talk, ma'am. Thank you with this. Once again, I congratulate the Department of Biotechnology for taking so much of efforts in conducting this webinar series. So that is the series on fermentation technology. So definitely this will be useful to our students and also to the community in large. Thank you very much. Thank you for this opportunity.
Thank you, ma'am. I take immense proud privilege to introduce Dr. K. Veena Gayatri among all of us. Veena Gayatri ma'am has got a teaching experience of 10 years and research experience of 7 years, and she has completed her PhD from Anna University Center for Environmental Studies on the topic studies on the degradation of phenolic compounds by a moderately hydrophilic halophilic bacterial consortium. Her area of interest are environmental biotechnology, wastewater treatment, integrated method for recalcitrant removal. She has also got a patent to her credit titled A Method for the Degradation of Chlorophenols by a Halotolerant Bacterial Consortium, which was awarded to her in the year 2017. She has completed various projects sponsored by Tamil Nadu State Council of Science and Technology, Tamil Nadu Science and Science Council Technology Student Scheme during the year 2018-19, DBT sponsored Full Scope to conduct workshops. UGC minor research projects was awarded in the year 2016. The awards that are bestowed upon her are UGC Fellowship for Meritorious Student in Science, Awarding Agency UGC Year 2005, Scientific Excellence Awards, Awarding Agency Biozone Icon Awards Year 2018, and she's also won various Best Paper Awards in national and international conferences. She is currently guiding 4 PhD scholars and she has completed 52 postgraduate student projects. The number of book chapters published by her are 6. She has published 45 international publications in reputed journals with good impact factors. She has organized various national level conferences and workshops. The organized and conducted, sponsored by the DBT Fourth Scope scheme during the year 2018-19 is worth mentioning. She's been an expert in external expert committee for academic audit in Department of Biotechnology in New College, DG Vaishnav College in large number of years. She's been a panel judge for various events and she's also represented herself in Indian Science Congress Association Chapter 27 during the year 2019. Yeah, Dr. Veena Gayatri ma'am, I would like to invite you to share your expertise for today's session, ma'am. Yes.
Thanks a lot, Bharati ma'am, for that nice introduction. And I would like to thank the principal, vice principals of Guru Nanak College and Dean of Research for encouraging me to present a lot of webinar series conducted through Guru Nanak College. I think yesterday I have been talking to you on. You're able to hear me, sir? Everyone is able to hear me?
Yes, ma'am. Yes, ma'am.
Yeah. So we are actually I spoke to you on media formulation, different methods used for formulation of a media, and different components which are present in media formulation. Today, I am going to give you an insight on what a metabolite is, a production of a metabolite, and a little of what mass transfer is. So both of these are big topics. So I'm just going to give you a gist of what metabolite production is and how a mass transfer occurs inside the fermentation unit. First, let's start with what metabolite production is. So I'll just introduce to what are metabolites, a small history of it, and primary and secondary metabolites. When you look at metabolites, these are actually intermediates and products of their metabolism of a microorganism. Typically, they are very small molecules and they have various functions. Mainly, they are categorized as primary metabolites and secondary metabolites. When you look at the history, metabolism as such is actually a Greek word. Metabolismos. This is actually it is how the metabolism was called. But as in French, it is called as metabolism. Okay, what metabolism is inside a microorganism? It actually says the metabolic pathway which is actually involved, and we examine the individual metabolic reactions which is involved with the biochemistry and molecular biology of a microorganism. When you look at these metabolites, they are produced by the microorganisms as intermediates or as products during their metabolism. When you look at the metabolite, it is restricted to small molecules. Example, in case of primary metabolite, they are actually involved in the normal growth, development, and reproduction. Example, like carbohydrates, proteins, lipids, nucleic acids, all these come under primary metabolites. When you look at secondary metabolites, they are actually not involved in the process of any ecological function. Example, like antibiotics, mycotoxin, but both primary and secondary metabolites are produced by the microorganisms. Okay, why are we so much interested towards these primary metabolites? Primary metabolites, as I told, they are involved in normal physiological processes. Hence, they are called as central metabolites. And it is very essential for the growth, development, and reproduction of the microorganism. Example, they include alcohol such as ethanol, lactic acid. An example of that is Aspergillus niger, which is an important organism in an industrial microbiology where you use it for a mass production of citric acid, one of the most important primary metabolites. Next is secondary metabolite. As the name describes, it is secondary. They are not involved. They are still the small molecules. They are not involved in any ecological function, but they do have lot of application in industry. Example, in case of antibiotics or mycotoxin, they are divided based on their chemical structure, their chemical composition, solubility with the solvents, and they have their own pathway by which these secondary metabolites are produced by the microorganisms. But why does secondary metabolites are of importance or they are of no use to microorganisms? Example, these secondary metabolites are actually produced at the late growth cycle, that is the end of the stationary phase. It is otherwise called as the eriophase. These secondary metabolites are structurally diverse and most of it are low molecular weight compounds, and this actually serves to increase the fitness of the microorganism and they decrease the fitness of other microorganisms present in the medium. So the metabolites play a crucial role in regulation and the process of sporulation in case of fungi. But there are two main differences which you have to look at when we are looking at a primary or a secondary metabolite. Primary metabolites are carbohydrates, fats, and proteins, which are main components of microorganisms. But when you look at secondary metabolites like alkaloids, tannins, resins, gums, latex, all these are secondary metabolites. Sometimes these secondary metabolites are poisonous in nature. They themselves can kill the microorganism or even the targeted microorganism which actually produces this. But this doesn't happen with the primary metabolites. Okay, let us look at, I'm just going to take one example, mycotoxin, which is a secondary metabolite. This is produced by the fungal organism. They are not necessary for their growth or in the developmental stage of the fungi. But some of these have very severe effects on animal, plant, and microbial system. Say, for example, the most important fungal food poisoning is due to the presence of these myco intoxication. The mycotoxin, myco means fungi. So the toxins which are actually released from them. So the symptoms of mycotoxicosis is that it is depending upon the type of mycotoxin it is produced and how much concentration it is present in the food, and as well as the age, health, and sex of that individual. So each of them will have a different way in which they react towards this mycotoxin, which is a secondary metabolite. Okay, I'm going to go into detail of primary metabolite production. I'm going to explain you what antibiotics are a little bit, and then go on for a simple production of antibiotic. Okay, what this term antibiotic actually means. Actually, this was coined by Watman in 1942. They are secondary metabolites which are produced by microorganisms. When I'm saying about antibiotics, most of them know that it is not only for bacteria. They are produced for as antibacterial agents, antifungal agents, and antiprotozoal agents. And some of these are also anti-tumorous and have antiviral properties. That is why they are called as antibiotics. It is used for treatment of any type of infection caused by the microorganism. It can even include fungi or protozoa. Most of the people think that only bacterial infections can be treated by the antibiotics. But actually, it is a general term which is given for fungi, protozoa, and other microorganisms as such. There are different types of antibiotics, but the broad classification is penicillin, cephalosporin, streptomycin, and griseofulvin. We are going to look at penicillin in detail. This secondary metabolite is actually not required for the growth of the fungus, but this fungus has a biomass in their growth phase called as a tropophase. They accumulate largest amount of penicillin during this phase, and they produce this with near exhaustion of the carbon source. What I mean by this statement is the media, when it is exhausted with the carbon source, during that time, penicillin will be produced by the microorganism. And you can see that high levels of glucose can repress the production of penicillin. And cephalosporin, I forgot to tell you one thing about penicillin. Penicillin, if you can look at, penicillin is a beta-lactam antibiotic. One of that kind is cephalosporins. These are semi-synthetic beta-lactam antibiotics, which are derived from Cephalosporium acrimonium. That is the microorganism which is actually involved. Other than these two antibiotics, there are alkaloids. Alkaloids are actually a group of chemical compounds which are actually contain nitrogen atom in them. Besides the carbon source or hydrogen molecule in the alkaloids, they also have sulfur, bromine, or phosphorus, which is actually present. These alkaloids are considered for many production like ergotamine, oxaline, and paramine. So these are all the products which are currently in the field even today where it is used for industrial production. Okay. Now I'm just going to give you an outline about microbial culture techniques for metabolite production. So what are all the culture techniques which has to be followed? Excuse me, sir. I'm hearing a disturbance from the other side. Can the other person mute? Mahindran, sir? Yeah, thank you.
Ma'am, I have muted, ma'am. You can proceed, ma'am.
Yeah, thank you. Thanks a lot, sir. So, I'm going to talk to you on what microbial culture techniques are for the metabolite production. Carefully look into the techniques which are going to be employed. I'll be going stepwise. Okay. So here, when I want to develop a industrially important microorganism, the first thing I have to do is isolate that culture. And most of the time, when you isolate the culture, you have to preserve that culture. Once it is preserved, you have to check whether there is going to be good implication in the industrial point of view. The first stage is the screening of microorganisms. That is, very potential microorganisms are isolated either as pure cultures or as mixed cultures. I think everyone knows that term, what pure culture is, or as a consortium. I just repeat, pure culture of the organism is nothing but the single culture from a single stain. But if I am talking about a mixed culture or as a consortium, it is a mixture of culture where many stains can exist in the same culture. So I should determine for my industrial production whether I am going to go for a pure culture or as a consortium. And this is determined by their desired reaction towards a desired product. Based on my product, I select my microorganism, whether I need a pure culture or I need a mixed culture. So to isolate, I must carry out the process in a very economical way. When you're looking at industrial production, it is as not just as 1 gram or 2 gram production of a product. We are looking at gallons of the product using so much of raw materials. Since it is has to be taken care very economically of selecting the culture. So there are different ways in which in way you can select the cultures. I'm going to explain this in 4 different methods. How can I isolate the microorganism of having a desired characteristics? First thing is I do through liquid culture technique. The method of enrichment is called as where I'm able to achieve an organism that is able to grow in my liquid culture. Second thing, I will be discussing about how I'll do the enrichment culture using a solid media. In that, I can see the viable cell count. The third one, I will be talking about the isolation methods which can be used for desired characteristics. I will not have any idea of the organism, but still I can isolate the organism. And the last one is the screening methods. I can see how many number of microorganisms are going to be screened for a particular type of product. First technique, enrichment culture technique. I think you can just see in your left hand side, I have given you a diagrammatic representation of how do you do it. See, I think every one of us would have gone through the first experiment in our isolation technique, a serial dilution. Same way, when I am enriching a liquid culture, either it is going to be from a natural source like a soil, sewage, or a contaminated soil, or a water contaminated one, or from a saline soil, whatever may be the content, I have to put it in a growth medium where I will be able to allow the organisms to grow in the liquid medium. Once there is going to be incubation period which is over for the liquid medium, I can do the serial dilution. So I can go from 10 power minus 1 till 10 power minus 10, and when I am plating these cells, I have to go for consecutive plating, which means if I am starting plating methods, it should be like -3, -4, and -5, or else -4, -5, and -6. I can't jump in between my dilutions. In that case, when I am jumping between my dilutions, I will not be able to achieve what type of growth of my organism it is. The same thing I have given in the words on the left hand side. So you can see that this is a liquid culture enrichment where I take the soil, add it to the conical flask, keep it for incubation for the required medium to grow, do a serial dilution, and then plate the cells. And consecutive plates are going to be prepared, and I count the colony forming units per ml and identify the microorganism based on their difference in their morphology. The second method, it is enrichment culture using solidified media. I have taken an example of isolation of a nitrogen Utilizing bacteria. Okay, how should I isolate a nitrogen utilizing bacteria? I want to check whether the organism is able to grow or not. Hence, I go for mineral salts medium. I just want to explain here what this mineral salts medium is. You can call it as M9 medium, M13 medium. There are so many references which are available on a mineral salts medium. But the simple MSM medium means I want only my required microorganisms to grow in that medium. You can see that the first flask is being supplied with mannitol as the source. So this mannitol will allow only the nitrogen fixing bacteria to grow, and there will not be any ammonia or nitrate or any organic nitrogen which is present in that. Now what I do in the same way I did for the liquid culture, I distribute that and add the soil and keep it for incubation. Once I keep it for incubation aerobically, the organism will start growing in my flask, and I am going to do the serial dilution and I am going to plate the cells. You can see that, for example, if I want to isolate rhizobium, a most important example of nitrogen fixing bacteria, we have to check the pH of the medium in which I am going to grow that, and I am going to plate the cells. You can see that the colonies will be formed only in the ammonia containing plate. Based on this, I can screen the purpose of nitrogen fixing bacteria. This is the second technique used for enrichment culture. Okay, the third one is I'm not going to think about any desired characteristics. Say, for example, I want to know about an organism which produces an antibiotic, the one which is going to produce a growth promoter. In that case, I take pool of microorganism, just do a spread plate or a pour plate and check whether the organism is able to grow or not. This example is called as statistical stepwise discrimination technique. It is otherwise called as SDA. This technique involved based on the nutritional requirement, you can classify the organism and take up the organism for further study. This is the third method of isolating the industrially important microorganism. And the last method is the screening method. Okay, this is said to be the most precise method where I will check for the desired identity of my microorganism. And for example, I will check only for antibiotics. If my targeted compound is antibiotic, I will screen only for antibiotic containing compound. And this will help me to isolate the potential producers of the organism, grow them in a liquid culture and in the cell-free broth, and plate them and check. So this is again a screening method by which I can isolate the microorganism of my interest for industrial production. Okay, first thing I have done the isolation. Now, once I have isolated the culture, it is very important step which I have to take. Really, a lot of caution has to be taken for preserving my culture. A simple way of preservation, I think most of us we carry out in the lab, is to store that in the refrigeration temperature. Say, for example, you keep it at 5 degrees Celsius or you keep it at minus 20 degrees Celsius. But what happens in the refrigerator? It is a liquid media, so it will get dehydrated. So I can't use liquid media. Another way of storing it is in the agar slopes. I can have it as a slope or a slant or as a stab. I can have any of these methods to store my cultures. Other than this, I can have liquid nitrogen to store my cultures, where the vials are filled with the microorganisms and it is stored with the liquid nitrogen, and I can keep it in the deep freezer. And the last one, most important part is the lyophilization. I think that it is otherwise called as freeze drying. I think everyone is able to observe here. There are a lot of wires which are being placed, which is being capped, and you can see a cotton which is inserted in the center. In this case, I can see the microorganism. They are in the dehydrated form. Along with addition of a adjuvant, I convert the microorganism into a form of a powder. And this is done with the help of the instrument called as lyophilizer. So what happens is at the end of the lyophilization, I can see that all the organism which I am in the foresee in the form of agar or in the liquid media, the pellet is completely turned in the form of a powdered form. Whenever I want, I can make multiple copies of these lyophilized culture, break open the lyophilized culture, revive that in the media, check for the growth of the organism, and further use for the industrial production. So these are all the methods which are actually given for preservation of the interested microorganism. Initially, I told you about how to isolate and then the screening method. Now I told you how to preserve these cultures. Okay, I have preserved these cultures, but some of the microorganisms will work, some may not. So I have to do some improvement in my industrial microorganism. So how do I do that? Selection of induced mutants in the improved levels can be done, especially for primary metabolites. It's little easier than the secondary metabolites. And you should see to it that there should be some modification which has to be done with their permeability process. And when you isolate such mutants, we should not produce any, that should not, the organism should not produce any feedback inhibitors or repressors. If there is going to be any inhibition or repressors which are going to be produced in the medium, that itself will kill the microorganism in the fermentation media. So isolation of the mutant is recognized with the presence of inhibitors and repressors, has to be experimented in the lab and then taken for the higher scale up. Okay, how do they do this? The improvement is done with the microorganism by certain methods. Selection of a stable stain, and you should see to it that the selected stain is resistant to infection. It should not get, like no, it should not lose its power when the mother microorganism is going to play a role on it. And the organism should have a non-foaming activity. I think yesterday when I was telling you about what is foaming, anti-foaming, I explained you how it will affect the fermentation broth. So you should take an important step to know that some of the microorganisms, say especially bacteria, bacillus, will produce larger amount of spores and larger amount of foaming. But in that certain strain, say for example, Bacillus subtilis, that organism, they don't produce so much amount of spores and they don't produce foaming. So you should select the microorganism in such a way that they have resistance to the foaming and as well as they produce less amount of foam in the fermentation media. And you should select the organism based on their morphological identification. You have to know whether they can survive even with very less amount of oxygen tension, because in between the fermentation process we don't know when the oxygen levels will go down, because there will be a lot of competition between the microorganism during their generation itself. So they should be able to tolerate a low oxygen tension in the medium. And you should see to it that they don't produce any undesirable products. The production state should not produce higher amount of undesirable product, but it should produce good fermentation products, which is our target. So that is how you improve the and modify the parameters of the industrial state. Okay. Yesterday, again, I told you about what an inoculum is. I have to repeat it here. These are certain criteria for taking up the microorganism from the lab scale to a fermentation vessel. Inoculum is actually defined as the population of microorganism or the cells that is introduced into the fermentation media, or it can be to any other suitable medium. You can just see the pictures which I have given above there. It is inside the laminar flow. You can see a madam who is actually plating the cells with the help of the inoculation loop. At the end of the inoculation is over, after 24 hours of incubation, you can see the organism is grown as a pure culture. So the inoculum needs to be optimized. You have to check the viability of the organism in a agar plate. And this, their performance can be varied based on different types of mutation, where they carry out the mutation based on DNA recombination, radiation methods, or by chemical addition. So this is where inoculum plays a major role in introducing into the fermentation media. Okay, now I have developed an inoculum. Yesterday I told you about what are the steps involved in inoculum development. But I am going to differentiate for bacteria how it works and for fungi how it works. Let us take bacteria. In case of bacteria, we have these vegetative cells and as well as spores. But what you have to do is, once you know they are as lyophilized cultures, I think the diagrammatic representation, you can see the first one, it's a lyophilized culture. You have to mix the medium in sterile water, tap water, or a sterile saline, and then add it in the logarithmic phase into a conical flask, which is containing a flat bottom flask that has a medium that will enhance the growth of the bacteria. And you have to make multiple copies of the same cells in the test tubes anytime you will require the microorganism during the fermentation process. In case of fungi, especially for non-sporulating fungi or actinomycetes, you can see that the hyphae is used, and inoculum size is very, very important. You should see to it that the size of the inoculum is always maintained from 0.5 to 5% of your media volume. So you can see that 20 to 200 folds the inoculum will rise during the fermentation process. So this is where inoculum is actually going to be added inside a fermenter. Yesterday, Bharati ma'am would have explained you about different types of fermenter. In all the fermenter, you will have a port in which a inoculum can be added in the aseptic condition. And after the addition of it, in between every cycle, you have to take up the organism through the withdrawal port, and then you have to culture the organism in the lab and check whether the viable cells are present or not, and morphologically see how it looks like. Okay. Another most important criteria for taking up an inoculum is take a healthy and an active inoculum. That is, they have to be in their exponential phase. Actually, this is determined by this formula. You can see that lambda n is equal to -1 divided by mu log. Where n is, this is very simple. Probably it is just the generation time what is actually given, the doubling time which is represented by 2n, where small n is nothing but the size of the inoculum, and expected log value of the initial inoculum is again n. So this formula will help you to determine whether your inoculum is present in their active stage or not. Okay, I think I discussed on the 3 or 4 slides before on this. Hello, sir. Hello.
Yes, ma'am.
You're able to hear me now, sir?
Yes, ma'am. Perfect, ma'am.
Okay, sir. Okay, sir. Because I'm not. Usually there was a sound which I can hear. I'm not able to hear that. Okay, sir. Thank you, sir. Thanks a lot,
Perfect.
sir. So next is optimum size of the organism. Say, if I take an inoculum, if I take one inoculum starting from the slant or from the stock culture, cells will undergo for around 20 to 50 generations. As I discussed with you earlier, you take up a microorganism which has to be added to the fermenter. Either it is a bacteria, or it is a yeast, or it is going to be a mold. 0.5 to 5% of medium volume has to be added. And this is going to increase to 200 folds before the fermentation ends. So low inoculum value will always take for long fermentation cycle, and this will lower the productivity of your product. And if you say there's going to be high density, okay, I want to add more amount of inoculum, this will again lead to a criteria what is called as inoculum effect. And you can see that in that case, the microorganism will start competing with each other, and they will not have sufficient amount of carbon, nitrogen, other sources in the medium to compete. So you have to have optimum size of your inoculum, either as a bacteria or as a fungi or as an actinomycetes. The importance is very much given to the size of the microorganism, which has to be calculated based on the quantity of the medium which you are going to take. Okay. Next, most crucial point in the fermentation is the contamination process. This is a main problem with microorganism, as they will lead to a very low productivity by killing the microbes which are present along with them. Each of them will be competing with themselves for producing the product and for their metabolism. So the inoculum preparation is very important, and you have to check the contamination in between every given cycle. Okay, this is again determined with the differentiation dn by dt is equal to kdn. where you can see that the number n, I'm very sorry that I'm not able to work out this in a board and explain, because that is actually a crucial way to explain how the derivation and kinetics are. So if you look at the n, that is the number of viable cells which are present, and you can see the small t, that is the treatment time at which I'm going to do the sterilization, and kd is the reaction rate constant. So this has to be calculated for every cycle of my fermentation process. I'm just giving you an example what happens in when there is going to be a contamination. Example, canning industry, if you take Clostridium botulicum. This is actually a bacteria which is a obligate anaerobe. This can grow in sealed cans and they produce heat resistant spores and they produce deadly toxins. But the same bacteria, if you see in sausages or in jams or in jellies, or if it is added with a sugar content, they will not have any contamination. So imagine a single bacteria is going to produce toxin in a closed can, but in case of sausages or jams where sugar content is there and which is stored at low temperature will not have any effect. So it is very important to check the targeted organism whether it is able to grow in the fermentation media or not. Please kindly look at the top of the slide. The first one, the yellow color organism, that is the kind of organism which I am inoculating before I am putting inside the fermentation media. And the next day, if I'm going to have the fermentation cycle, for the first cycle, going to see the aspergillus, I can see the yellow color is completely converted into green color, and the center is completely green. And throughout that, I am able to see the mycelial mat, which is spread with white color patches. And when I am observing that under the microscope, I can see the conidiophosphores that is actually exposed. But the second cycle, which is given in the pink color, if you could just see, the same organism is able to show some contamination. In this case, I have to completely throw away the media, whatever I have prepared, the inoculum, whatever I have prepared, and the steps, whatever I have taken to complete my fermentation process. So, contamination is very important to be taken care when you start the fermentation. I have discussed enough about culture media yesterday. I told you about water, carbon source, nitrogen source, vitamins, and minerals. These play a really a very important part in media design, and you can see that how much amount of carbon and energy source you're going to provide as nutrients. That is the kind of product, carbon dioxide and water and heat and biomass which is going to be generated. So culture media formulation and design plays a crucial part in industrial production. Okay. Other than this, there is something called as where I can improve my strain by having a classical method of preparation. See, today only we have recombinant DNA technology being involved where I can just use restriction enzymes, cut the DNA at different sites I want. I can insert whatever the targeted gene of my interest. But a long time back, they do what is called as selection technique where you allow the microorganism to be in an environment for a very long time. So what happens is it goes for classical mutation on its own. As I told you, a microorganism when I'm going to inoculate into a media, either into a liquid media or into a solid media, will undergo several generations, 20 to 50 generations it will undergo. So for example, I'm going to work on a textile effluent treatment where I want to remove the textile dyes which are going to be present. So initially, I just take a flask, add the amount of dye, expose the microorganism, allow it for 50 generations. Now, what will happen is, without any gene manipulation, at the end of 50 generation, I will have mutants of organisms which can withstand the dye and they can degrade the dye. This is called as the classical stain no, selection method. So this is an outline of how you can improve the strain of the microorganism, getting a maximum yield. Okay, I think I just told you about that. You start with the mutation treatment. See, this can be just UV mutation, or you can expose it to chemicals and allow the spores to grow. Once a spore suspension is ready, you allow them to get plated onto a agar medium. I think when you plate it onto agar medium, nearly 200 colonies are going to be produced in the media. Always go in for higher quality and higher quantity of your microorganism during the initial stages of industrially, isolating industrially import microorganism. Always prepare in duplicates, triplicates, and quadruplates. You make the stock culture of it and do the preservation method. Have the preserved culture as such. Now, other than that, on your left hand side, you can see that the same spore suspension is used for the development flask. You check the growth of it, how it is able to grow, and each of this flask has to be plated against, and you can check what is the mutation which it has undergone and check for the inoculum preparation. You should not just do it with one single flask and decide I have got the best organism. Always check minimum of, if you have 20 flasks, minimum 10 flasks should answer the mutation what you have created for the targeted product. Okay, what actually they do today in recombinant DNA technology to improve the stainness? Say, for example, in vivo for Penicillium chrysogenum, they are using the recombinant DNA technology to check the penicillin. They undergo the parasexual cycle. And for example, in case of fungi, yeast, and bacteria, they do protoplast fusion. And in vitro conditions like streptomyces, actinomycetes, and filamentous fungi, they are using the methods for improving the fermentation products, like especially for antibiotic production. Okay, this is the natural variant which I already explained to you. Microorganisms undergo a slight genetic change with every cell division. So after a several amount of cell division has occurred, you can see that there is a range of genetic structures which has been changed in them, and you can take these natural variants for the industrial production. Okay. Other than these, there are some other methods like immobilization of cells. Say, for example, some of the microorganisms they don't have so much amount of no production as a in the media. So what do you do is you attach them into a solid substrate. Say, for example, you can allow them to produce the product or as an inhibitor. This process of immobilization can be done in different ways, like a physical method or chemical method. Especially in industry, they employ carrier binding method, entrapment method, and cross linkage method. So this process helps you to get a high molecular weight hydrophilic polymeric gel. Like I think everyone would have heard about sodium alginate, agarose. All these are simple polymeric gels in which you can immobilize your cells and inoculate the organism in that, allow it to grow, and that can be used for industrial production. Okay, now everything is set. Your medium is ready, your inoculum is ready, and you have standardized the organisms. You have checked for the contamination. You transfer the inoculum to the fermentar vessel and then go for the scale up. So yesterday, ma'am has explained you on how the valves work. So please check the valves properly before you start the fermentation process, and always the inoculum has to be transferred at a aseptic condition inside the fermenter. It could be noted that 5 to 10% of total culture volume has to be maintained, and you should have sterile disposable syringe which have to be used at the suitability. And the vessel is actively aerated, and you should see to it that there is a chance of getting contamination in the vessel. So the syringe is quickly withdrawn immediately after the inoculation. And as it is a silicon tube, it will reseal immediately, and then the scale up process starts. You start from a small system and then go on for a large system and start checking up the scale up with the help of oxygen supply and with your heat removal and other key factors which was explained to check for the scale up process. So most importantly, any industrial production you check, they do what is called as antimicrobial susceptibility test. Okay, this is actually used to check the proper inoculum preparation is done or not, whether it is accurate or not. So they check the inoculum purity and inoculum size. This is otherwise even called as quality control. You check the presence of the microorganism, their antimicrobial activity, how it is processed. Okay, so again, I am showing you a outline of how the preserved cultures can be taken up from the seed fermenter into a large scale fermenter, and then you separate the biomass and the downstream processing. I think today or tomorrow you'll be having a talk on specifically on the downstream processing. Inoculum purity, I have already explained you, but one most important point which I want to tell you again here is any inoculum, if you take 4 to 5 colonies of similar morphology has to be taken. And when you're taking in a liquid media, say for example in peptone water, or it should be suspended in normal saline. These are the two important criteria which are actually used before you check the inoculum purity. Okay, I'm just giving you a outline of how our penicillin production is done. Okay, I think you can just see in your right hand side the penicillin chrysogenum, the microorganism which is actually produced in the agar plate. It is an SDA plate, Sabadose dextrose agar. See, penicillin is said to be the first antibiotic which was developed, and this was the antibiotic which has actually saved so many of them during the World War II. Okay, so the composition of the fermentation media is given here. You can see the carbon and nitrogen sources which are added, like corn steep liquor, cane, molasses, glycerol, other minerals which are added along with water, the media which has to be added. So once you prepare the inoculum, say you take the primary source of the inoculum from the soil, one or more stages you have to plate the organism in the solid media and check whether it is growing or not. Once it is grown in the solid media, turn them into the shaker flask. Into the shaker flask, whatever is going to go has to be checked for the seed stage. Say, for example, you have zero point 0.5 to 1 meter cube per the seed stage. Now increase it to 10 to 20 meter cube. That is again increased to 125 to 250 meter cube. Like that, you go in for a bench top reactor. This you allow the penicillin to produce for 40 hours. Once the antibiotic is completely produced, it is extracted with organic solvent, especially butyl acetate is used, and you can precipitate the antibiotic using the potassium salts. So that is how you recover the penicillin, and then you convert them into the vacuumized form as lyophilized form, and then you prepare them as tablets. With that, I just finished with different types of inoculum development, primary and secondary metabolite, and the production. I'm just going to introduce to you to the mass transfer. This actually requires a lot of time to explain, but I'm going to put it in very small words where everyone can understand what actually happens in as mass transfer inside the fermentation broth. Okay, what exactly is mass transfer? Actually, transfer of the mass means it is the chemical species which are being transferred. It can be into the system or out of the substance. Both are possible. See, I'll give you some simple examples. Liquid converting into gases. Okay, that is an example of mass transfer. A diffusion, which is occurring from a higher concentration to lower concentration. Just your RO unit at home, where you have your water, which is actually moving through the RO membrane from a higher concentration to a lower concentration. That exactly is diffusion. Okay, what are the other main things which we see in our house or during our course of day to know what mass transfer is? Evaporation in the form of drying, baking, frying, boiling, all these are mass transfer. Okay, other ways I should say diffusion. Say, for example, you have the cheese, you take it from the fridge, you keep it outside. For some time, you can see that there will be small salt deposits on the surface. Okay, you take the meat, you take it out of the smoke, and then you keep it down. For some time, the smoke is still there. That is again diffusion. And say, for example, you prepare a meat, there is a curing solution after some time comes out of the meat. That is again diffusion. But what is not mass transfer? Moving a fluid from one place to another is not mass transfer. The chemical species which is actually moving from one end to another, and that is going to nullify the whole unit. Okay, that is mass transfer. I just want to define what mass transfer is. When you take a system, it can have two or more components whose concentrations vary from point to point, and this has a natural tendency and it minimizes the concentration. So this is actually mass transfer. Probably it is little difficult for you to understand the definition, but when I'm explaining you what mass transfer is with a diagrammatic representation, you will be able to appreciate it better. There are two ways in which you can call mass transfer. One is called as molecular diffusion, or it is just called as diffusion, as it is a random collision of molecules from a concentration gradient. But it is not always heat transfer or conduction. It can be by any method. Then you have what is called as Eddy's diffusion. This Eddy's diffusion occurs during the mixing or dispersion, and probably even during the agitation which is caused inside the fermenter. So what happens in Eddy's diffusion is the mass is transferred into the form of finite parcels of fluids, and there will be a momentum which will be created. And you can see that the rate of diffusion varies for gas, liquid, and solids. I am going to give you a simple example of what the diffusion occurs. When I am telling about the types of mass transfer, there are two types of mass transfer. One is called as molecular diffusion, other one is called as convection mass transfer. I'll explain this in detail. Just a small idea. Just take up a beaker, add water to it. To that, add a cube of sugar. Without you don't disturb that for a longer period of time, at the end of 10 minutes you can, or 10 minutes or even 7 minutes, you can see that there will be complete diffusion of this sugar into the vessel where it is completely dispensed. After, no, I don't want to wait for 7 minutes. I want to activate that process. So what I do, I just insert a spoon inside and add the sugar and mix it well. Now what happens is I am actually mixing it in such a way that the diffusion is occurring faster. If it is automatically occurring on its own with the molecular diffusion, the chemical movement, it is just called as diffusion. But if I'm going to add with a mechanical force to it, where I'm going to move it with the help of the spoon, still the dispersion occurs. It is called as convection mass transfer. So this is a simple way of explaining what a mass transfer is. So I am stirring the water with the help of the spoon. That is actually creating a force, and that leads to the bulk water which is actually dissolved with the sugars. Okay, are we thinking only here it is occurring? Imagine what is occurring in your lungs. At the surface of your lungs, if you say you have air which is outside and you have blood which is inside your body, imagine the air which is outside is having the oxygen. And this oxygen is going to be at a very high concentration, and they will have a very low concentration of carbon dioxide. But inside your blood, if you say, they will have very low concentration of oxygen and they will have high concentration of your carbon dioxide. This is again an example of mass transfer. And on other way, oxygen enters inside your blood cells and carbon dioxide comes out of the your lungs. Both the ways diffusion is occurring, and this is carried out with the help of your lungs, which is a mechanical way of pressing both oxygen which is pressed inside your cells, and as well as the carbon dioxide which is pressed outside the cells. So diffusion is a net transport of molecules from region of higher concentration to a region of lower concentration. This is what is called as molecular diffusion. I have another way to explain you in a simple way of what diffusion is. Just look at this diagram. You have A and B. Initially, when I am having A and B, I am having a partition of a black color, no, a line on the center. On yellow color and red color is completely separated, and they are separated from each other. Now I am removing that line. What happens to that? I can see that from the higher concentration, the B is a dye which I have added to a media. So what happens is once I have taken that line, black color line, there is a diffusion of the dye from B to A. At the end of the diffusion, it becomes a single yellow color line or a mixture of orangish color which I'm able to get from yellow and red. This process is called as diffusion. This is what happens inside the fermenter. In case of eddy size, why I'm saying about all this is all this plays a crucial part role in a fermentation technology when you're running a fermentation vessel. So there is a lot of turbulence which will be created by the bubbles which are going to be formed. The turbulence is the word actually means that there is a lot of kinetic energy which is actually involved during the rotational movement. And in case of turbulence, it is called as eddies. Okay, all the bigger eddies will get converted into smaller forms, and they form what are called as smaller eddy size. So this also plays a very crucial part in case of your fermentation process. Okay, how do I determine this? This is determined by Fick's law of diffusion. That is, the rate of the molecular flow is directly proportional to the difference in the concentration by the membrane of the dimension. I'm explaining this again in detail here. You can see the Fick's law of diffusion. I'll just explain you what are all the parameters. No need to just get worried about the equation there. You can see that JA is nothing but the diffusion flux or the mass flux. How there is going to be a movement in the perpendicular direction of the media into the system. And you have NA, the rate of mass transfer, where small a will show you the area across the mass transfer. And you can see that DAB will give you the diffusion coefficient, and the C is nothing but the concentration gradient. I believe I showed you a diagram where there is a movement of red of B which is converted to A, and they become orange in color, and that is determined by this Fick's law of diffusion. The second method is, or the second mass transfer is called as convection mass transfer. You can see that mass transfer takes place only with single phase or either to or from a phase boundary. As for example, sublimation of naphthalene, that is, it is actually in the solid form, but it never gets converted into a liquid form, but it goes to the air. Okay, this is an example of a convection mass transfer. Mass transfer even takes place when two contacting phases are going to be together, like an extraction or an absorption method. And this is determined by the formula given here. No need to bother about the formula too much, but it is just the mass transfer coefficient which has to be calculated, and they determine what is the convection mass transfer. Most importantly, I just want to talk about what the last part of the talk on gas liquid mass transfer. That is, every microorganism if you take, they will have a specific oxygen requirement. Only based on that, they develop themselves into the fermentation media, they grow, they metabolize the components which are present and produce the product. Okay, what is this gas liquid mass transfer? I just want to show you a small diagrammatic representation here. You can see there is a gas bubble on towards your right hand side. Okay, number one. Now what happens here is the gas bubble moves towards the interface. The first dark color line which you see is the gas liquid interface, which is number two. From that two, it moves towards the liquid film. That is, the microorganism is actually somewhere else, and you see the gas bubble in the media. Now, this liquid media, from that, two things can happen. One is it can be as just as an individual cell. Just see number four, that is nothing but the media. Bulk liquid is nothing but your media, the fermentation media. So two things can happen. Some of the microorganism can be as individual cells where they will undergo the attachment to the oxygen content which is going to enter into the individual cell. But sometimes you can see as a cell, as a clump, where they will form a biofilm. So in that case, many microorganisms will join together and they will form a biofilm. So it will take some more time for the microorganism to take up that oxygen inside the cells and take up the oxygen for their supply. Imagine just a gas bubble from which oxygen has to be supplied to the microorganism, either as an individual cell or as a film. It involves 8 steps. That's what I have given here. The first step is transfer from the interior of the bubble to the gas liquid interface. Second thing is they move across the gas liquid interface. Third, they diffuse themselves relatively in the stagnant liquid surrounding the bubble and transport through this through a bulk liquid, and from that the stagnant liquid surrounds the cells. Then there is going to be a movement across the liquid cell interface, and if it is going to be a flock or it is going to be a clump or as a biofilm, they will have as a solid particle. So diffusion inside the solid particle is still more difficult than a individual cell. That is how oxygen will be transferred from the gas bubble, the aeration which you are giving in the medium, to the microorganism which it has taken. So this transport will occur through the cytoplasm on the site of reaction. So with that, I end my presentation on mass transfer and metabolite production. Thank you. I'll be very happy to answer your questions. Sir?
Yes, ma'am. Yes, ma'am.
Yes, sir.
Aarti, ma'am.
Thank you, Dr. Veena Gayatri ma'am, for your very lucid and informative session. Now I call upon Dr. Mahendra Kumar, Assistant Professor, Department of Biotechnology, Guru Nanak College, to take over the discussion session.
Yes, ma'am. Thank you very much, ma'am. Dear participants, we request you to post your questions and comments in the chat box so that it will be easy for ma'am to answer. One minute.
Okay. They have asked, Srinivasan sir has asked about fermentation. The inoculum should be from master mother or mother culture. Okay. Srinivasan sir, when you're starting a fermentation, no, the inoculum is actually from your seed culture. So, for example, you're starting with 250 ml flask. You start getting the process of from the 250 ml flask. You go for 500 ml and then go for 1000 and 2000. So, in that case, the inoculum is already being developed. As I showed you in different places of my presentation, you have to have your seed culture which is going to be ready throughout the lab culture. And once you know the organism is very active, it is completely able to participate, those inoculum are again taken for the industrial scale. Okay. How is medium affecting the mass transfer? Yes. See, medium will surely play a crucial role in affecting the mass transfer because, as I told you about the foaming properties of a microorganism, as well as the components which you're adding in the medium. Say, for example, you have so many raw materials which are going to be used, especially sugars. Sugars will help the microorganisms to grow, and as well as they will create larger amount of frothy nature that will affect the growth of the microorganism, and it will inhibit the production also. So you have to be very careful. And there is something called as I told you, certain formulas. I was not able to give you the whole outline about it. There are crucial formulas which has to be determined, especially which is used for molecular diffusion and as well as for convection mass transfer. This stoichiometric calculation has to be calculated before it is taken to the fermentation unit. Only then, if you are able to work out with your lab scale, you can take that for the pilot scale study. Okay, there is a possibility. Yeah, Surya Arun sir, he is just asked about whether there is a possibility of microbes to get mutated after inoculation. Yeah, yes, Surya Arun. It is a very narrow chance, as I discussed this in a larger time, saying that microorganisms have the tendency to mutate. But when you are selecting the strains, you have to be very careful whether they are stable strains. They have to be stable strains for several generations, not just for one or two generations. 50 generations of one microorganism has to prove the same production. Say if I'm going to take an antibiotic penicillin, it has to give the production for 50 generations. Only then I can use that for fermentation, or else the whole lot of your fermentation process will go for a failure. Okay, Rahul is asked about during mass transfer, will other factors be affected or not? Yes, Rahul, during mass transfer, it is not only going to contain your product formation, the inoculum will be affected, and your mass transfer will hinder the product formation, byproducts form, and sometimes even the inhibitors will go in for a rise as the mass transfer is going to be enhanced in your fermentation media. So we have to reduce the mass transfer occurrence inside the fermentation broth before it is taken for higher scale up. Oh, thank you, Lokesh sir. I want to have a lot of other things also. I actually contain a lot of things. Next one, does the mechanical injury of the plants increase the secondary metabolite? Yes. So see, during secondary metabolite production, it is primary metabolites are actually said to be simpler. You can easily produce primary metabolites. Secondary metabolites, as I told you, they are non-essential for the microorganism and mostly produced only at the end of the stationary phase. So you have to be very critical about knowing the growth phase of your bacteria, actinomycetes, or fungi. If you don't know at which stage your secondary metabolites are produced, it will surely affect your fermentation process. And there are some secondary metabolites produced by plant tissue cultures, but that itself can be enhanced in different way of adding the inhibitors and precursors. Neela Devi has asked, diffusion and convection are different in mass transfer. So how do you calculate energy transfer in the fermentation? Okay, that's a very good question. Neela ma'am, how it actually goes is, first thing you have to look at it is a molecular diffusion. That is the first diffusion technique which I told you. Because convection mass transfer is going to occur based on the mechanical stirring. So based on agitation, the aeration you give, the other methods which are going to be followed. But when you look at molecular diffusion, that plays a crucial role. I think I told you about the diffusion flux. Please refer Schuler and Kargi. That's the most important book which is giving on mass transfer. They will clearly explain about what is solid liquid transfer, liquid liquid transfer, gas liquid transfer. But first thing is to calculate molecular diffusion. If you are satisfied with molecular diffusion, that itself will take care of conventional mass transfer. How to measure actual cell size in case of viscous fermentation broth? Okay, Manthan Kulkarni. In case of viscous fermentation broth, most importantly, if it is bacteria, please don't go in for viscous fluid because viscous microorganism producing the cell mass will surely affect the product because the downstream processing will become very difficult. But in case of fungi, there is no other option. We can go for viscous fluids. There are viscometers which are available. We can do viscometer testing before it goes to the final scale. Viscometers are available. It's a simple method. People may think it's very difficult. Viscometer is just a 100 rupees glass syringe. If you can just calculate, take the broth, dilute it with broth or with sterile distilled water, you can calculate the viscosity of your microorganism. Once you know the viscosity, based on that, you can add the broth into the fermentation vessel. So that way you can prevent the viscosity problem with the bacterial cells. Few points on most effective methods of preservation culture method that is used worldwide. Yes, I would really advise you, it can be a simple microorganism which you have isolated. Kindly go in for lyophilization. And lyophilization, you have to do it with some adjuvants. Say you can even use a milk powder, skim milk powder can be used as adjuvants. Just take that powder, sterilize it. There are different ways in which you can add these adjuvants, add your microorganism to it, and then go in for lyophilization. Say lyophilization will just cost 200 or 300 rupees. You will be working for 6 months for getting a culture. But if you don't lyophilize it, the end of the microorganism will die. And there is a lot of chance it mutates. So kindly go in for dried samples as lyophilized cultures. Okay, how amoxicillin is identified in surface water? Antibiotic additions are there are a lot of techniques which you can predict antibiotic production which is there in the surface water. But when you look at surface water, it is not in every water you can see amoxicillin. Especially when you go into a place where pharmaceutical industries are there, there you get so much amount of amoxicillin which is there. And there are a lot of protocols. It is there in APHA. Okay, it is a wastewater treatment analysis. You can check for both potable water, surface water, which is available, where you can check APHA protocol, standard protocol. E-book is available in the net. You can download that and you can check for the analysis. Oh, thank you, Neelama Devi. Thank you. Thanks. I can share the references. I have just followed Schuler and Kargi. The most important book is in bioprocess. So Schuler and Kargi is available. It is microbial bioprocessing book, which you can just refer. All my notes, what I am prepared for this class, is from Schuler and Kargi. Okay, what is distinct properties of primary and secondary plant metabolites and their proportional presence in all plants? And then one thing which I want to tell you, according to these primary and secondary metabolite, especially with plants, is there's an experimentation process. Say, for example, you have the metabolite which is going to be detected in the leaf, and you select the plant. And especially when you're starting with any primary or secondary metabolite, we start with a reference. So minimum have 10 to 12 reference material with you before you start for a production, an industrial production, and check all that for the production in your lab scale. Once you have the proportionality of the production, then you can go in for the at the different scale. And I would like to make a comment here saying that every plant, say for example, it may be a small secondary metabolite like auxins. Every plant has a different way in which they express in their tissue culture. So there is a different level of production of these metabolites. So you have to standardize the method in the lab and then go in for the final scale up. Yes.
Ma'am, that's all. All the questions you have answered, ma'am.
Oh, thank you, sir.
Thank you very much, ma'am, for the clear presentation and the very interactive question and answer session. Now,
Thank
I
you, sir.
request the head of biotechnology, Dr. Bharti Ravikrishnan, ma'am, to say a few words.
Thank you, Veena Gayatri ma'am, and thank you, Mahendran sir. I take this opportunity to thank our speaker, Dr. K. Veena Gayatri, Assistant Professor, Department of Biotechnology, Stella Maris College, to share her expertise on the topic metabolic production and mass transfer. I hope everyone will agree with me that her talk was very informative and she has delivered a very detailed information about the inoculum development and a clear and a very simple method of explaining the mass transfer. I take this opportunity to thank our General Secretary and Correspondent, Mr. Manjit Singh Nair, in supporting in all our department endeavors. I thank our Principal Dr. N.G. Raghunathan for his critical suggestions. I also take this opportunity to thank our both our Vice Principal, Madam Dr. L.R.S. Kalanidhi ma'am and Dr. Gunita Arunchandok ma'am for their suggestions and support in conducting and organizing this webinar series. I thank all our participants for their enthusiasm in participating in this live YouTube recording. I thank my department colleagues, Dr. Mahendra Kumar, Mr. Amal Ludraj, Dr. P. Mahalakshmi ma'am, and all my students in bringing this fifth session of webinar today. Thank you. Now over to Dr. Mahendra Kumar for a few more instructions.
Thank you very much, ma'am. Dear participants, thanks for your participation in the last 5 sessions, and we welcome you to the 6th session, which is to be held at 3 o'clock today, and the links have been shared. And the resource person for the 6th session 6 is Professor Dr. Ramalingam, Professor, Center for Biotechnology, Anna University, who will be enlightening us on the topic different modes of cultivation of microbes for fermentation. So please join us for the 6th session at 3 o'clock sharp. The YouTube links have been shared to you by email. We have also posted the YouTube link in the respective WhatsApp groups. Please join us. Thank you for your participation. Thank you very much. Kindly fill up the feedback forms, the links which have been posted in the chat box. Thank you very much.